Today we will explain a review in Science Translation Medicine, titled Neuroinflammation and Astrocyte Perspective. Astrocytes are the most abundant glial cells in the central nervous system CNS, where they perform extensive homeostatic functions, such as buffering excess neurotransmitters, regulating synapses and blood-brain barrier BBB function, and supporting other CNS resident cells. In the context of neurological disorders, many of these homeostatic functions become impaired. Furthermore, the activation state of astrocytes influences the pathogenesis of multiple sclerosis (MS), Alzheimer's disease (AD), Parkinson's disease (PD), Huntington's disease (HD), and other disorders. Cell-cell interactions serve as central modulators of astrocytic homeostasis and disease-associated responses. This review highlights the intricate interactions between astrocytes and other CNS resident cells, as well as CNS infiltrating cells, exploring their potential therapeutic value in inflammation and neurodegenerative diseases. During homeostasis, astrocytic subtypes exhibit genomic and functional characteristics related to their location and interactions with other cells in the CNS. However, during CNS inflammation and neurodegenerative changes, astrocytic cellular states correlate with the activation or suppression of specific genomic modules in response to disease-specific stimuli. For instance, Hassel et al. Identified two astrocytic response states following lipopolysaccharide administration. One astrocytic state is characterized by enrichment of the metalloproteinase inhibitor TIMP1, indicating neuroprotective and antioxidant functions. The other state features gene expression associated with interferon signaling, angiogenesis, and antigen presentation. This highlights astrocytic heterogeneity under both CNS homeostasis and pathological conditions. Importantly, over 97% of astrocytic feet contact at least one blood vessel, enabling astrocytic membrane channels, transporters, and receptors to sense and respond, not only to intrinsic CNS signals, but also to circulating molecules. In this context, cytokines, metabolites, and other cellular products, along with environmental factors, such as environmental chemicals and microbial metabolites, collectively modulate astrocytic responses related to antigen presentation, co-stimulation, and apoptosis. Astrocytic reactivity and associated signaling pathways are differentially regulated across various CNS diseases. For example, multiple studies have highlighted the importance of the STAT3 signaling pathway in reactive astrocytes. Selective inactivation of STAT3 in astrocytes reduces amyloid plaques and improves spatial learning and memory, divided by PS1 mouse models. Furthermore, in CNS inflammation, Ilmenus-6 modulates glial neurotoxicity via STAT3 signaling. However, in traumatic injury, reactive astrocytes form a barrier around CNS lesions, thereby confining inflammation. Astrocyte-specific STAT3 inactivation impairs motor recovery and significantly increases pro-inflammatory gene transcription in spinal cord injury indicating the role of STAT3-controlled astrocytic products in CNS repair. Thus, STAT3 in astrocytes context specifically control CNS inflammation and neurodegeneration. Now, let's talk about astrocytes' interaction with neurons. First, astrocytes regulate synaptic transmission and circuit function. For example, they participate in tripartite synapses functional units formed by astrocytic processes and neural synapses, enabling efficient bidirectional astrocyte-neuron crosstalk. Astrocytes support synaptic function by secreting synaptic factors, phagocytosing structural synapses, and buffering synaptic glutamate. In neurological diseases, these and other homeostatic astrocytic functions involved in synaptic support become impaired. Second, astrocytes exhibit metabolic coupling with neurons. For instance, astrocytes metabolize glucose into lactate, supplying it to neurons to support their metabolism. This metabolic crosstalk between astrocytes and neurons is disrupted during inflammation, thereby amplifying neurotoxicity. Similar observations in HD, AD, and PD underscore the significance of astrocyte-neuron metabolic decoupling in CNS inflammation. Moreover, Mounting evidence points to astrocyte-driven neurotoxicity. Activation of NF-bin astrocytes promotes production of neurotoxic nitric oxide No. Moreover, microglia-induced reactive astrocytes present in HD, AD, PD, and MIS brain samples promote neuronal death by secreting neurotoxic fatty acids, suggesting shared astrocytic pathological drivers across diverse neurological disorders. However, disease-specific triggers exist for astrocytic activation associated with distinct transcriptional and cellular responses, suggesting multiple astrocytic states with neurotoxic activity can be induced. This further indicates that even within the same disease context, astrocyte-driven neurotoxicity may involve multiple mechanisms. Finally, neuroprotective mechanisms driven by reactive astrocytes are also emerging. Episcopo et al. Propose reactive astrocytes as a source of WNT1, which profoundly influences dendritic neuron development and recovery. Researchers observed astrocytic WNT1 expression in response to MPTP-induced dendritic neuronal degeneration. Astrocyte-derived WNT1 binds to FRIST receptors and kidney and dopaminergic neurons, promoting their survival, repair, and neurogenesis. Oligodendrocytes play an active role in CNS inflammation. They express multiple cytokines, immunomodulatory factors, and their receptors. For example, Kim and colleagues reported that astrocytes promote TNF release from microglia, which binds to TNF receptor when expressed on oligodendrocyte precursor cells, and induces cytotoxicity in a contact-dependent manner. Upon CNS injury, OX are recruited to the lesion site in response to Ilmenus-1 and CC chemokin ligand to CCL2 produced by astrocytes. Once recruited to the lesion, OX differentiate into mature oligodendrocytes under the influence of ciliary neurotrophic factor CNTF. Interestingly, downregulation of the NRF2 pathway in astrocytes promotes oligodendrocyte survival and myelin regeneration. Furthermore, recent reports indicate that during this pathogenesis, oligodendrocytes disrupt the blood-brain barrier BBB by downregulating tight junction proteins. This BBB disruption is driven by oligodendrocytes competitively enveloping blood vessels with astrocytic infeed. Given the age-related impairment of myelin regeneration, determining whether astrocyte oligodendrocyte interactions are disrupted during aging is crucial. This represents a key point for developing therapeutic strategies to promote myelin regeneration, particularly in neurological disorders, where myelin regeneration defects and disease progression are associated with aging, such as MIS. Ilmenus-33 secreted by astrocytes promotes microglia-dependent developmental synaptic pruning, highlighting the importance of astrocyte microglia crosstalk in neural circuit development. Figure 1. Moreover, microglial factors modulate astrocytic pathogenic activity during CNS inflammation. For instance, Bezzi et al. Demonstrated that microglial TNF induces SDF-1CXER4 signaling, driving astrocytic glutamate release and promoting neuronal death. Conversely, astrocytes can also regulate microglial responses in pathological CNS contexts. B4GALT6-driven lacquer production induces astrocyte secretion of GM-CSF, modulating the transcriptional responses of microglia in CNS infiltrating monocytes. Similarly, the SIGMAR1ERE1XBP1 axis in astrocytes not only drives intrinsic astrocytic responses promoting CNS pathology, but also drives pathological microglial responses through uncharacterized astrocyte-microglia communication mechanisms. Astrocytes constitute the primary component of the blood-brain barrier BBB, forming the glial boundary membrane barrier surrounding peripheral cells, endothelial cells, and the basement layer that restricts leukocyte entry into the CNS. However, BBB disruption and dysfunction can facilitate leukocyte invasion into the CNS parenchyma. Under such circumstances, astrocytes are among the first resident cells encountered by infiltrating T cells, regulating not only T cell migration, but also T cell activation. Additionally, astrocytes serve as major sources of chemokines such as CCL1, CCL2, CCL20, and CXCL10. NF drive an expression of CCL2 and CXCL10 in astrocytes recruits pro-inflammatory T cells and monocytes to the CNS, promoting NIS and EE pathology. Furthermore, activation of rank receptor activator of NF on astrocytes, a rank ligand expressed by Th17 cells, triggers CCL0 production, thereby facilitating the rare recruitment of effector T cells during CNS inflammation. Moreover, the relationship between astrocytes and T cells is bidirectional, as T cells can also shape the astrocytic response. Astrocytes express receptors for IL-17 and GM-CSF, which are produced by pro-inflammatory Th17 cells implicated in the pathology of NIS, EA, and other diseases. Th17-derived IL-17 was initially thought to upregulate ACT1-dependent pro-inflammatory cytokine GM-CSF and chemokine CXCL1, CXCL2, and CCL0 production in astrocytes, promoting leukocyte remobilization to the CNS and EAE. However, subsequent studies revealed these IL-17 effects primarily involve glial cells, expressing neuroglial antigen to NGG2. Thus, understanding of IL-17 signaling in regulating astrocytic responses remains limited. Nevertheless, GM-CSF was found to promote MUFK-divided IMAT to dependent pro-inflammatory astrocytic state differentiation in NIS and EAE, while simultaneously restricting expression of the T cell apoptosis inducer trail in astrocytes. The recruitment of peripheral mononuclear phagocytes plays a crucial role in central nervous system inflammation. In NIS and EAE, astrocyte-derived CCL to re-recruits pro-inflammatory monocytes to the central nervous system. Deficiency of CCL2 in astrocytes reduces the recruitment and polarization of pro-inflammatory monocyte-derived macrophages associated with demyelination and neuronal death. However, astrocyte-derived CCL2 can also recruit macrophages to promote tissue repair, indicating that astrocytes can modulate pro-inflammatory and reparative mechanisms in a context-specific manner. NK cells circulating through the meningeal circulation induce trail expression in quiescent astrocytes by producing and thereby causing apoptosis in pro-inflammatory T cells expressing death receptor 5. Notably, NK cells circulating through the meninges acquire the capacity to produce IFIN in intestinal tissues in response to signals provided by the commensal microbiota. These findings identify mechanisms within the gut central nervous system axis for regulating inflammation, revealing not only the potential role of the microbiota in the pathology of neurological disorders, but also offering exciting opportunities for therapeutic interventions. B cells play a crucial role in central nervous system homeostasis and pathology, as underscored by the success of B-cell-targeted therapies in NIS and other neurological disorders. Within NIS plaques, astrocytes produce B-cell activating factor BAF, which is essential for B-cell development, survival, and antibody production. Secretory factors produced by human astrocytes promote the survival and activation of B-cell subsets, including memory B cells in patients with secondary progressive NIS. In NIS and stroke, astrocytes secrete CXCL12, which promotes plasma cell maintenance and survival, indicating astrocytic roles in controlling pathogenic B cells within the CNS. B cell products also influence astrocyte function and survival. For example, antibodies reacting with aquaporin drive opticospinal demyelination by inducing astrocyte dysfunction and death. All in all, cell-cell interactions play a central role in the functional control of astrocytes in health and disease. These interactions are disrupted in neurological disorders, suggesting that disease-associated connectomes involving astrocytes and other target cells may represent important targets for therapeutic interventions. That's all. Thanks for your listening.